Guys, today we'll study about meiosis 1 and meiosis 2 phases. We'll just continue from the interface first part and we'll study about the meiosis 1 first. Okay. After meiosis 1, we'll discuss about prophase 1 and the metaphase 1, then we'll continue about anaphase 1 and the lastly we'll do about telophase 1 and cytokinesis. Then we'll step towards meiosis 2. We'll discuss about prophase, metaphase 2, anaphase 2, telophase 2 and mainly cytokinesis. Okay, yeah, now you know that in meiosis 1 what happened is separation of the homologs, separation of homologous chromosomes. I got you remember that. And now in meiosis 2 what happened is separation of sister chromatids, I guess. Okay, they get separated and resulting in the 4 haploid cell. Now let's study this. So first of all, let us talk about some interface. Okay, what previously happened. You see this is a cell. Okay, at a time of interface, centrosomes are present with two centrioles pair, these ones, and a ring array of form around them, which is called esters. Okay, then also the nuclear envelope is present there with the nucleolus and nucleus inside it, and chromosomes are there. They are not duplicated. Sorry, yeah, they are duplicated, but are not divided. Not divided. Okay, at this point of stage, you know I have colored the homologs as red, and blue as the other homologs. I told you about homolog in the previous video, I guess. First and second, homologs are the pairs of the chromosome run from the each parent, which have the same size, shape, and carry the same gene in the same location, but can have different version of alleles or example of these gene. Let me just write for you. Wait. Okay, here it is written now. Okay, for example, one can be mother, one can be father, but they have same gene location. You can say locus point. That's why they're homologs. Now just let's dive into the meiosis one part. So for meiosis one, what's the first thing? I guess you remember P match structure: prophase, metaphase, anaphase, telophase, and then cytokinesis. So what is first? First is prophase. You guessed it right. Prophase is the longest and the most complex phase of then the mitosis. Prophase is first is longest, okay, more complex than mitosis. It is unique for what? To meiosis. Now different thing will happen here. So first of all, see, this is a cell here, okay? This is a cell. These are what? Centrosomes. Like what you can see here, the centrosome have moved opposite to of the poles toward each cell. Opposite poles, they have moved to opposite poles, okay? Centrosomes. And now the spindle fibers, you can see the spindle fibers, they are forming. They begin to form. Okay, you can see the nuclear envelope here. It broke down. Like in the interface, it was clearly intact, but it broke down. And this process, it was generally the same as mitosis. Till this part, it is generally same. Okay, now what happened next is the chromosome get condensed, more condensed. Okay, they become shorter, thicker, and visible under the microscope. This condensation will continue till the prophase one, all throughout prophase one. Now next thing, new thing, you can see two chromosomes are here. They are what? Homologous chromosome. Okay, one from father. Let's say this one from father, this one from mother. Okay, now they align by gene. Okay, they align gene by gene here at this point. They overlap each other and meet at a point. Okay, this thing is called synapses. The connection between these chromosomes, the connection between these two chromosomes is called, or we can say the pairing, it is called synapses. At this point, they meet a point, it is known as loci, or we can say loci. This paired structure is called as bivalent or tetrad. Why? Because there are four chromatids here. One, two, three, four. Okay. Now let's talk about the crossing over. So basically, what happened in crossing over is, you know, this is a father chromosome. This is the mother. They're homologous. Okay. Let's name the sister chromatid as, like, this sister chromatid I named that is one. This is one dash. This is two. This is two dash. It is from father. It is from mother. Okay. Now what happened is, some part of father get cut and realigned to the mother part, and some part of mother and get cut and realigned to the father part, but at the same point. Okay. This thing is called non-sister chromatid. Like, one dash and two are the non-sister chromatid, like, because the father has uh, one and one dash, mother has two and two dash. Okay, these sister chromatid one dash and two, they are non-sister chromatid because they are not related to each other. That's why. Okay. Okay, now when they like crossed over, okay, it leads to what? Leads to genetic exchange of the genetic material. Now this is important part, because what it do, like, it's create the genetic variation, produces a recombinant chromosomes, and like, this thing, this key thing you've seen, like, it doesn't happen in the mitosis area. Okay. See now, I have written down for you also, like, the DNA, the chromosome, like, which were like shorter, it one like more thicker, visible under the microscope. Okay. Basically what happened, I told you, DNA get break down, okay, rejoins with the other chromatid, leads to what? Exchanging of genetic material, like here. And furthermore, it will exchange out further in the next phases. You will see that. Now, new thing, chiasmata, or we can say chiasma. Chiasmata. Chiasma. What does this mean? This is the like physical evidence of crossing over. We can literally see that in the each homologous pair, it has one or more chiasmata, which we can say uh one chiasmata here, one here, or we can say many more. Like, what does, like, how this help? It has to like hold the homologs together, like they are holding it together each other, like, by crossing over. And it also ensures the proper separation later on. Okay. Now what happened in the late prophase? Like, I just, before that, I will show you, written that also for you. Okay, till here. Now, like, what happened in the late prophase? See, now here a key thing happened is, the microtubules. Okay, they attach to the kinetochores. Each homolog has one kinetochore at its centromere, but it was different in mitosis. Why? Because in mitosis, the two kinetochores of sister chromatids are fused together. Okay, for example, this one is a chromosome. It has a kinetochore, but like, it was fused. But in meiosis, it acted as a single, like this. So one kinetochore here, one here. Okay, they act as a single kinetochore in this one, and which, like, this leads to ensure, like, the sister chromatids like move together and homologous chromosomes separate, not the chromatids. Now, spindle fiber, like, later on what happened, well, the spindle fiber begin like moving homologous pair, they are directed toward the metaphasic plate, then the axial alignment happen in the metaphase one, blah blah blah. Okay. Now let's talk about the metaphase one. Yeah, written this for you also. Yeah. Now what you can see here, you can see the pairs of the like homologous chromosomes, you can say bivalent or tetrads, are arranged at a metaphasic plate or metaphase plate, equatorial plane. This thing, it is known as what? Equatorial plane. all the homologs, they are arranged here. You can see here. Okay. Now you can also see like one chromosome faces one pole. For example, this is north pole, this is south pole. Just to we can say, yeah, north pole, south pole. Okay. This chromosome facing this pole, this chromosome facing this pole. Okay. The opposite, which faces the opposite pole. Like this orientation is random. You can see here. Now let's talk about the kinetochore placement. Now in kinetochore placement, you can see that, both sister chromatid of one more homolog. Okay, this sister chromatid I can say, and this sister chromatid I can say. Okay, this is one homolog as number as one. Okay, now they are connected to what? Kinetochore microtubules from the same pole. This is the same pole. They are connected to this. See this line. You can say this microtubule is connected to this sister chromatid and this sister chromatid also. Okay, and the same as the other section. This sister chromatid and this sister chromatid connected to the this pole via microtubule. Okay, in mitosis, sister chromatid attached to opposite pole. Okay, for example, first one will connect from this side. Okay, and second one connect from this side, which is opposite. But here it is done in same pole. Okay, now why this arrangement is like important, because like it ensures the separation of the homologous chromosome in the next stage. It maintains the sister chromatid together and like leads to the reduction in chromosome number 2 and 2n. Also written down for you. I'll provide you the notes also. Yeah. Now let's turn to our anaphase one. Now what you can see here, what you can observe. Take a time. What you can observe here, the breakdown of sister chromatid cohesion along the arms. Sister chromatic cohesion, like sister chromatid like join like this, which was done by a cohesion. It get break apart, but cohesion is only removed from the arm, not from the centromere. What happened next? This leads to what? Separation of homologous chromosome, but the genetic material was exchanged. Okay. Next thing. What happened after separation of the homologous chromosome? Okay, once the cohesion was broke. Okay, each homologs get separate from this partner. Okay, now the spindle apparatus pulls one homolog toward one pole, this homolog toward one pole, and one homolog toward this pole, other pole. This helps to what? Cause reduction division. I can say 2n 2n, diploid to haploid. But you can also see here the key thing that like sister chromatid cohesion remains at the centromere. Okay, sister chromatid like do not get separated. They move together at one unit toward the same pole, which is a key thing, and it is different from the mitosis. You know, now I've written down for you. Now let's talk about the difference from anaphase 1 and anaphase 2, anaphase in the mitosis. So like what is like separating here? The separation of homologs we can see in anaphase 1. In anaphase, it is separation of sister chromatid. The sister chromatid gets separated. And centromere cohesion, yes, it is maintained in the anaphase 1, but it is lost during the anaphase in mitosis. Like that's why like it is called like 2n 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 to 2n. The diploid doesn't changes in mitosis, but here it is changed 2n 2n. The chromosome number remains the same, but here it get reduced. Now we know like the homologs get separated now, separated. Okay, sister chromatid is remained till now. Okay, then the both of the chromosome, homolog chromosome, are at the opposite side of the cell. Now what will happen next? You guessed this right, cell division and telophase, which also means cytokinesis and telophase. Now let's toward it. You can see the uncontinuous formation of cell. No, the two cells are forming here. The nuclear envelope is forming. Okay, each half of the cell get at its haploid structure. Gets its haploid structure. Why? Because the diploid shifted to haploid as a chromosome, like the pairs got broken in half, and these chromosomes are still duplicated. You can see that. But but in this thing, each chromosome consists of two sister chromatids still. Okay, one or both chromatid may contain the what regions of the non-sister chromatid DNA, one from father, one from mother. You can see that. Okay, this is happened. Why? Because of the crossing over in the prophase one, and these chromatids are called recombinant chromatid. Okay, now let's talk about the cytokinesis. So cytokinesis, what happened in cytokinesis? It also occurs at the same time as the telophase one. The cytoplasm is dividing. You can see that it is in half, forming two haploid daughter cells. Okay, and each daughter cell is haploid, contain duplicated chromosome. That's it. Now let's talk about the difference between the animal and the plant cell. In animal cells, a cleavage furrow is formed. Let me just show you. I think you remember that. Like suppose this is an animal cell. Okay, okay. There's a thing called this line of thing is called as cleavage furrow. In this animal cell, you can see the line is forming from the middle. This thing is called a cleavage furrow. It is done from outside to inside, outside to inside. Now in the plant cell, a cell plate is formed, like which breaks the cell into from inside to outside, like this. That. Now let's talk about the general behavior of the chromosome. Uh, After the telophase one, in some species, the chromosome get decondensed, the nuclear envelope reforms. Okay, or sometimes in other species, like the cell move directly from the meiosis two without the full nuclear like reformation. And also we also know that like in the no DNA replication will like between happen in the meiosis one and meiosis two, because like the meiosis 2 start with the haploid cells and the chromosome cell consisting of two sister chromatids, and that's why I written this one down for you. Okay, now allow heads to us to the meiosis 2. Now here we know that we have two new cells, each are diploid, oh sorry, haploid. You can see that like now the uh, now we'll discuss from two cells. Okay, two perspective here. Now you can see that both of the cells, the spindle moves toward the opposite poles. Okay, the spindle apparatus forms. It's a new thing. Again and again, the centrosome moves toward the opposite poles. Microtubules like beginning to like organize here. And also this stage is similar to the prophase of the mitosis, but the cells are haploid, not diploid. One key thing to note is like, cells are haploid, not diploid. Okay, yeah. Now let's talk about the chromosome structure. Each chromosome is still composed of two sister chromatids. Okay, the chromatids are joined at what centromere. Okay, no DNA replication occur before this stage. The chromosomes are already duplicated now. Now let's talk about in the late prophase 2. What will later on happen is, the spindle microtubules, they will attach to chromosome. These microtubules will attach to chromosome here. Okay, and the chromosome are moved toward the metaphase 2 plate, metaphasic 2 plate. Now the key difference between prophase 1 and prophase 2 is like, pairing of chromosome. It is like present in the prophase 1, it is absent. Why? Because like in prophase 1 the crossing overs was done, and synapses was done there also from like at that time they were deployed, and they have only one cell. Now they have two cells, no. So that's why. Now also about the crossing over, it occurred in prophase 1, it did not occur in prophase 2. Chromosome number is not same, it was deployed earlier, now it is haploid. It is similar, like key question, like does it similar to like mitosis prophase 1? No. Why? The joining and crossing over occurs there first of all, and it's for deployed. But prophase 2 is more like mitosis, as a number remains same. Now now again metaphase 2. So what you can see here, the positioning at the metaphase plate, the chromosome aligned at the metaphasic plate, equatorial plane again, but this time they are with recombinants. Okay, like this time the chromosome lines up like individually, not in homologous pair like in metaphase 1. You can see they're not in pair right now. Earlier it was like this. Okay, now due to the crossing over the meiosis 1, the sister chromatids are not generally identical. They are not same. See here, each chromatid may carry different version of, or we can say combination of alleles from the father side, we can say, or alleles from the mother side. They are both different. Sister chromatid are not same, like which contributes to what genetic variation in future. Two cells still haploid. Okay, now about the kinetic or attachment, now here the key thing you have to note that is, see, kinetic or the sister chromatids, you know they are attached to the microtubules from the opposite pole. For example, suppose this is north pole, this is south pole. Okay, you can see this chrom- uh, this chromosome and this centromere, it is directly from the side of north pole, but it is directly attached from the centrosome from the south side. It is opposite side. you know Now this arrangement will what happen, if this attached from here and this side attached from here, opposite side, later on what happen is separation of what sister chromatid gets separated from the what at centromere location. Now they are attached at centromere, they will what get detached from it. This is difference from like metaphase 1, because why? At that during that time sister chromatid were attached to the same pole, but in metaphase 2 they are attached to different poles. This leads to what separation of sister chromatid we can see in anaphase 2. I've written this for you also. Now let's talk about anaphase 2. You can now see here the cytoplasm get like elongated a little bit. Now also you can see there, what is happening, key feature. Now the proteins like which are held in sister chromatids together, they separate. Okay, they are broken down at what? At the point of centromere. For example, this is a chromosome, and these parts are the recombinants. Okay, wait, this is a chromosome. Okay, these part are the recombinant. What happened is, earlier we know that earlier the chromosome get broken like this. Okay, at this point. But now in this stage, in anaphase 2, what happened is this: the chromosome breaks apart from the middle at the centromere location. And how it happened, you know, this centromer location, one of the chromatid going that way, opposite pole, or we can say one of the chromatid going to same pole, like this. The cohesion, like which were like there, like to hold the chromatid together, gets broken down here. Now what happened is, when the chromatid is gets separated, each chromatid separated from the individual chromosome, they will become what? New chromosomes, individual chromosomes. Okay, these chromosomes are pulled by the spindle fibers toward the opposite poles. This movement one guided by the like, spindle apparatus, and like it is very similar to mitosis. Okay, it is very similar to the phase of the anaphase of the mitosis, because like it was occurs, but like the key difference here is like it this occurs in the haploid cell. The chromatids are genetically different from the mitosis. That's it. Now why the anaphase 2 is important? Basically, it ensures the equal distribution of the chromosomes. Okay, it leads to formation of the haploid cell with single like unduplicated chromosomes here. And now it's complete the separation process which like started in the meiosis 1. Now we'll talk about telophase 2, the meiosis and the cytokinesis at last. Now what happened is this. Okay, suppose these are two cells here now, both haploid still. Okay, in these cells the cytoplasm started to get divided. Okay, what are the main thing is, like you can see the nuclear envelope reforms. Now one can also see the nuclei reform at the like each pole of the form. Okay, the chromosome begin to decondense again. They become long, thin, and thin. Long, wait, long, thin chromosomes. This marks the end of meiosis 2. Okay, now nuclear envelope reappears, and yeah. Now let's talk about the cytokinesis later. Now, at this point of time, cytokinesis is also occurring at the same time. The cytoplasm is dividing. Okay, what will happen later is, result of getting four daughter cells, one, two, three, four, which are again what? Haploid in nature. The chromosomes are what? Like they are unduplicated single chromatids, chromosomes. This is the key thing to note. This is different from the telophase one. Like like the chromosomes were duplicated, and now they are also like genetically unique, because like these daughter cells are genetically different from one another. Okay, they are different from the parent cells. This is the reason, because the crossing over occurs in the prophase one, independent assortment of the homologous chromosomes, and the random segregation during the meiosis 2. That's it. The four cells form here. Now let's like just like talk about the final outcome, what happened later in final outcome. Number of cells, 4. Chromosome number, still haploid. Structure of chromosome, unduplicated. Genetic makeup, all different. They're not from the same as parent. That's it. Meiosis is over. Now I've written notes for you also. From now on, I'll provide you notes for the lecture also. It will be at a like link at the bottom.